Welcome to today's lesson, KCSE 2017, Paper 3, Biology. Let's see what it contains. In question 1, you are provided with the following materials and reagents. A straight portion of raw banana labeled D, 2 petri dishes, a scalpel or a razor blade, 2 beakers containing liquid E and F, measuring cylinder, stopwatch or access to a wall clock. The means of labeling. Now in the procedure are told, label the 2 petri dishes E and F, place 30 centimeters cubed of liquid E into a petri dish E, and 30 centimeters cubed of liquid F into a petri dish F. Using a scalpel, prepare 4 thin, straight flat strips from the raw banana peel. Each strip should measure about 4 cm by 2 mm as illustrated. Now you are given a note. If you want to get a straight flat thin strip, remove all the banana flesh, leaving only the peel. Then immerse 2 strips in a petri dish E and the other 2 into petri dish F, and leave the setups undisturbed for 10 minutes. Now the questions. State your observation in petri dishes E and F after 10 minutes. Now up to now, and you have the observation you will see, you should be able to know the nature of the solution E and F. The solution E and F, now the observation is, in E, the strip curved outward, while F, the strip curved inward. Now up to now you should be able to know now the nature from the observation. Petri dish E is a hypotonic solution because the strip in E loses water, while in F gains water and become turgid. Yes. As I have said, account for the observation made in A, Roman 1 above. Liquid E is the hypotonic solution. Liquid E, water entered into, now this is you are accounting. Water entered into the mesocarp of the banana, into the mesocarp of the banana peel. By what? By osmosis. Therefore, this we are investigating the process of osmosis. Then the inner cell became turgid. The inner cell became turgid than the outer one, leading to the curvature, outward. Now the strip looked like this, peeled outward. Now this is the epidermis. Epidermis, you should be able to know, has a cuticle which is waterproof. Now it has peeled outward. Is the epidermis has peeled outward. Now this solution, we said it is a hypotonic solution because it increased in size and then pulled outward. In Petri dish F, the inner cells lost water by osmosis. The inner cell lost water by osmosis. They became flaccid or shrink. They became flaccid or shrunk, causing the inward curvature. Now it looked like this. Now this is the epidermis. The curvature is now inside. It has curved inwards. You see now the difference. In hypotonic, the curvature goes outwards, while in hypertonic, the curvature curves inwards. Should have the content in osmosis in cell physiology. Now in B, describe the nature of liquids E and F in relation to the sap in the banana peel used in this experiment. Now liquid E, we know then the nature is liquid E is hypertonic. Liquid E is hypotonic, while liquid F is hypertonic. Yeah. With reference to the observation made, compare the nature of the outer and the inner surface of the banana peel. Now the outer surface of the banana peel is impermeable. While the inner surface is permeable. The nature of the outer and the inner. In D, we are being asked, name the cell structure responsible for the observations made in this experiment. The cell structure. This is the key word. The cell structure. Cell structure is the cell membrane or plasma membrane or the plasma lemma. Explain how the structure named in D1 above works to bring about observations made. Now the response will be, it is semi-permeable. The structure is semi-permeable to allow selective movement of materials in and out of the cell. In question 2, you are provided with the following materials and reagents: 3 test tubes, dilute egg albumin, this is another name of protein, albumin of protein, access to dilute HCl acid with a dropper, and sodium hydroxide solution with a dropper. Then you are given solution P. Then you have two droppers, three 10 milliliter measuring cylinder, and use a stopwatch or a clock wall. And then access to water bath maintained at 52. When you see temperature maintained, in your mind you should think of the enzymes. In your mind you should have a copy of enzymes in this question. Label the test tubes A, B and C. Put 2 cm of the egg albumin in each of the 3 test tubes A, B, C. Add 1 cm of solution P in each of the test tubes. In the test tube A, add 2 drops of sodium hydroxide. In B, 2 drops of HCl acid. And in test tube C, add water. And then place all the 3 test tubes in a water bath for about 10 minutes. Now the observation in test tube A and B. In A, the content in the test tube turned colorless, or not turned, formed a colorless solution. While in B, content in test tube B was cloudy. Content in test tube B was cloudy or white precipitate or milky. Cloudy, white PPT, or it was milky. Account for the observation in one above. The observation in test tube A was, in test tube A we have seen we added sodium hydroxide. Therefore sodium hydroxide provided in A, sodium hydroxide provided in A above was suitable because the enzyme it was suitable because provided a suitable working condition for the enzyme, which was enzyme P. P was an enzyme on the egg albumin. Hence there was effective digestion. In test tube B, HCl, we added HCl. HCl provided was unsuitable suitable for the working of enzyme P. Hence there was no digestion, hence there was no breakdown, there was no digestion or breakdown of the egg albumin of the egg albumin. Yeah. Now, when you see an acid, HCl is an acid. This one denatures the enzyme. This one denatures the enzyme, while this one is suitable for action of enzyme. This is suitable for action of enzyme. Alkaline medium is suitable for the action of enzymes. Now explain why the investigation was carried out at the specified temperature range. That is simple because it was to provide an optimum temperature for the action of the enzyme of enzyme P. That is the enzyme P. What is the purpose of test tube C? That was only to control the experiment. You have seen enzyme in test tube C we added only water. Was to control the experiment. With a reason, identify the solution of P. P, we have seen that is the protein digesting enzyme. The protein digesting enzyme, because egg albumin is a protein in alkaline medium. Name the likely part of the human alimentary canal where the process in this experiment occurs. This experiment occurs in the duodenum because of the alkaline medium. Occurs in the duodenum. And you are told give a reason. This because it has an alkaline condition or medium. In question 3, you are provided with the specimen labeled H and K. H was the bean leaves, while K was the maize leaf. Now, specimen H is a complete plant, while K, not J, is a portion of the plant. Observe and answer the question. State 3 observable features between H and K. Now this is H, this is K. Now you have an idea of a bean seed, of a bean leaf. Now it has a broad lamina, and this has a narrow lamina, while has network veins and K head parallel veins. Also has short leaves, and K have long leaves. Explain 3 ways in which the stem or specimen H adapt the plant for maximum photosynthesis. H has a firm stem, to expose leaves for light or to light for maximum photosynthesis. Also, the stem are green that contain chlorophyll to trap light for photosynthesis. Also the stem has phloem to transport the product of photosynthesis. In 2, explain the ways in which the plant from which specimen K was obtained adapt to survival in its habitat. Has green leaves for photosynthesis. Also has many fibrous root system. For anchorage. Also have many nodes. Many nodes to allow growing or propagation of the plant. Explain the consequence of adding liquid F used in 1. Liquid F, we said that is the hypotonic solution. Therefore, the plant will lose water. To the soil, and by osmosis, and eventually wilt or dry up and die. That is the hypertonic solution. Lastly, state 2 ecological importance of specimen K in an ecosystem. The importance is it acts as a food for herbivores, reduces greenhouse effect, hence reduces CO2 in the atmosphere. That's all. Thank you.